Hello, everybody. Today, I'll present our work examining the cellular underpinnings of the human cortical connectome. I have no disclosures. I'll first briefly discuss two key properties of the functional organization of cortex, the macro-scale functional gradients and the large-scale functional networks. Then I'll present our latest work examining to which extent are these functional properties observed in vivo are mirrored in the spatial distribution of cells imputed from the postmortem tissue samples. Lastly, I'll discuss some exciting analysis suggesting that the functional network assignments can be predicted from the cellular profiles in postmortem tissues. As we learned from classical neuroanatomists like Brodmann, the brain cell types are not uniformly distributed across the cortex. Indeed, the Brodmann areas were originally defined and numbered based on the cytoarchitectural organization of neurons he observed in the cerebral cortex using the cell staining technique. That is saying, cell type is not a functional definition, but it can be a reliable biomarker or biotarget for the study of psychiatric population if only we could establish the stable alignment between functions and cell types. Talking about the function, initially formalized by Maslow and others, then recently confirmed by Margulis and colleagues in their study that decomposed the resting state functional connectivity into gradients, that the function of cerebral cortex are organized along two axes. The first axis, or gradient 1, spans between the unimodal areas such as the visual or auditory system, and the transmodal association areas, including heteromodal, paralimbic, and limbic cortex. Here to the left, you can see the transmodal areas colored in yellow are in the location that allow them to efficiently integrate the information from the nearby auditory and visual cortex. And the second gradient further separates the unimodal areas into the visual cortex and auditory cortex. They are positioned far away from each other so that we can't listen by our eyes or watch by ears. Also derived from the resting state functional MRI, vertices sharing similar functional connectivity to the rest of the brain can be grouped into the same network. Here, for instance, is the 7-network solution derived by Thomas and colleagues, then parceled in the Schaefer Atlas. Margulis and others found that these 7 networks are spatially organized along the above 2 gradients of the cortical function. So the 2 gradients and the 7 large-scale networks are the 2 major functional architectures we're going to target today. Again, as we know from Brodmann, that the brain cell types are not uniformly distributed across the cortex. Recent work by Anderson and others suggested that these cell type distributions have important implications for the study of in vivo brain functions. For example, the spatial variability in the bold amplitude is highly correlated with the relative distribution between somatostatin and the piva-Billman interneurons. These observations lead to a critical question for cognitive neuroscientists to answer. That is, how does the microstructure of a given patch of cortex influence its function? So in this project, we seek to investigate the extent to which cortical cytoarchitecture taken from postmortem tissue samples aligns to the in vivo functional organization of cortex. To address this question, our group and others have taken advantage of Allen Human Brain Atlas, built from postmortem brain of 6 adults. It provides whole genome microarray RNA sequencing data of bulk tissue samples across the brain. To estimate the abundance of cell types from the total transcriptions across those bulk tissue samples, we construct the gene expression signature of all the possible cortical cell types from single nuclei drop sequencing data provided by Lake and colleagues. Here, 17 subtypes of cells were identified. The single cell samples are obtained separately from the frontal cortex and the visual cortex. To control for the heterogeneity, we use these 2 samples as independent replicates to conduct the later analysis, named as Lake DFC and Lake VS. So, after the abundance for different cell types were imputed for all the bulk tissue samples in the cortex, we then compare them with the functional gradient and networks. The table on the right is color-coded by the Spearman correlation between the first principal gradient and the abundance of each cell type. And the number indicates the FDR-corrected p-values derived from the spin test that controls for the spatial autocorrelation. 3 out of 17 cell types survived the significant test for both independent replicates. Inhibitory 1 interneuron and excitatory 1 neuron, which are spatially anchored to the transmodal end of the first gradient, like the yellow areas, and the piva-Billman interneuron that's spatially anchored to the unimodal end of the first gradient, like the blue areas. To view the combined effect of the cells enriched in unimodal and the transmodal end of the gradient 1, we took the difference between the excitatory 1 and p-valvumen. Then we obtained a 0.557 Spearman's correlation. That is surprisingly good because the combination looks so much like the gradient 1. Encouraged by that, we did examine the multivariate cellular relationship with gradients and keep obtaining a correlation that's above 0.5, even after removing the cell types that are univariately correlated with the gradients. Given the limit of time, I won't go over the results here, but please come and chat me at the poster or check out the details in our preprint. Here's the QR code. Inspired by the patterns in the scatter plot like p-valvumen, here, that many cell types are actually preferentially enriched within certain networks. We then examine the heterogeneity of the cell type enrichment across different functional networks. For each cell type, take p-valvumen as an example. Its empirical abundance were aggregated within each one of the 7 networks. Its null distributions were obtained again by spin test, which permuted the empirical density for 1,000 times while controlling for spatial autocorrelation. The table is color-coded by z-score. Here, z-score measures how far away the empirical value is positioned relative to the null distribution. Blue indicate that the empirical value is lower than the median of null, and the red means it's higher than the median of null. So the number in the table indicates the FDR-corrected p-values for each z-score. These data suggest there may be a unique cellular profiles for some functional systems. To have a clear view of that, for each network, we visualize the relative enrichment of all the 17 cell types on a polar plot. For example, the somatomotor network. The z-score derived from the 2 independent replicates, like DFC and like VS, were overlaid on top of each other. You can see the pattern are pretty reliable between these two. The dotted circle here represents the median of null abundance for each cell type as a reference for enrichment. As you can see, in somatomotor network, p-valve and oligodendrocytes are very enriched, but excitatory 1 and inhibitory 1 are much less present. This pattern is almost flipped in the limbic network. You can see the excitatory 1 and inhibitory 1 are very enriched, but the oligodendrocyte and p-valvumen are much less present. Excitingly, we observed these visually distinct cellular profiles across each network, which suggests that the heterogeneous enrichment of each cell of the cell types could potentially serve as a fingerprint to identify the functional network. To test this idea, we trained a supporter vector machine classifier to predict the functional network from the 17 cell type abundances. The classifier was trained on 70% of the randomly selected parcels and tested on the remaining 30% parcels. This process was repeated for 1,000 times, which generated the blue distribution of the prediction performance for this classifier. And the similar process was conducted on 1,000 null cell type abundance profiles generated from the spin test. The null score of the prediction performance is colored as gray on the left. As you can see, in general, the classifier can predict the functional network each parcel belongs to from their cell type abundance significantly above chance. To take a closer look at how well the classifier can predict on each network, we calculated the recall of each network. Recall here evaluate the probability of classifying a parcel correctly, given that the parcel is from a certain network. From the plot, we can see 4 out of 7 networks can be predicted significantly above chance. There are somatomotor network, visual network, ventral attention network, and limbic network. So again, in our effort answering this question, to what extent does post-mortem cytoarchitecture align with the in vivo functional organization of human cortex? Well, 30% variance of the functional gradients can be explained by cell type abundance distributions. Embedded within this broad profile, we observed 4 out of 7 functional networks can be predicted significantly above chance by classifier trained on the 17 cell type abundance. These data suggest we may be able to establish the underlying molecular and cellular basis of the functional network changes we observe in patient populations. So I'll stop here and thank all those who support us on this cool works. And thank you, OHBM, for inviting me and funding the trip. For more details of this work, please scan the QR code for the preprint, and I'd be thrilled to contact and chat more with you. Thank you for your attention.